right. Like the other speakers, I'd like to start out by thanking the meeting organizers, especially David, for giving me this opportunity to present. It's also important to acknowledge members of my laboratory, both current members as well as past members who've done all the work about to present. So since starting my research program at NIH, we've been interested in exploring the role of p53 in regulating mitochondria, given the growing, increasing appreciation of the importance of mitochondrial metabolism in tumorigenesis as well as cardiovascular biology. So what I want to do today is share with you, like all the other speakers also, the thing doesn't move. Okay. So share with you our interest in trying to develop strategies for preventing cancer in Li-Fraumeni in LFS, or helping develop strategies for cancer prevention in LFS. And so as outlined here, I'll start out by sharing with you sort of some background published work where we've shown evidence of increased oxidative metabolism in Li-Fraumeni syndrome. And then go on to share with you some unpublished work where we've, using both genetic and pharmacologic means, using metformin, as Fasana very nicely outlined, have tried to disrupt mitochondrial function in an LFS mouse model and shown that indeed we can delay cancer formation. And then again, in collaboration with Fasana, we have looked at the feasibility of inhibiting mitochondrial function. So I hope my data can help complement some of the work that we're working together on in LFS patients. And along the way, I'll touch on a little bit more basic mechanistic study by which we demonstrate that p53 protein can physically move into the mitochondria. That's driven by active respiration, which is mitochondrial function, which appears to partition the activity of p53 between the nucleus and the mitochondria. And I believe this highlights the homeostatic nature by which p53 regulates mitochondria and also helps explain some of the LFS findings. So a number of years ago, we showed that p53 can promote mitochondrial respiration. And our work and those of others have shown that p53 does it through multiple target genes in the nucleus. And the two genes in particular that we've been interested in, SCO2 and TFAM. SCO2 is an assembly factor for the respiratory complex 4, which is also known as cytochrome C oxidase, that is critical for, that is the center of oxygen consumption in the eukaryotic cell. And also, we've shown that p53 can transactivate mitochondrial transcription factor A, which is a protein that's critical for mitochondrial genomic DNA transcription as well as its maintenance. And as shown here, p53 itself has been shown to physically interact, by work of other investigators, physically interact with proteins such as TFAM and polymerase gamma that are important for maintaining the mitochondrial genome, which is separate from the nuclear genome. It's a much smaller genome, but still, nonetheless, it's a separate genome. While it's important to advance our understanding of how basic mechanisms regulate the mitochondria of p53, it's also important to find its relevance in humans in order to see if we can try to advance human health as well. And so we addressed this question by turning to Li-Fraumeni syndrome a number of years ago. And this was work that was done through a Bench to Bedside award that we collaborated with Louis Strong, who really made this study possible by collaborating and referring patients to the NIH Clinical Center. And we wanted to be able to measure mitochondrial function directly in vivo in patients. And we took advantage of a technique. Sorry, it's really okay. We took advantage of a technique that was available within our institute. This is a phosphorus 31 MR spectroscopy technique we collaborated with people who had expertise, Bob Balaban and Lalita Lagala. And essentially, it's a real-time MRI scan, and we are imaging the phosphocreatine species, which is phosphocreatine is a high-energy molecule that's used for shuttling ATP that's required for contractile activity within the muscle cell. And what basically we do is we have the individuals, and I think some of the LFS patients have participated in this protocol, so they record this very well. But essentially, it's a foot pedal that is attached by a pulley system to a specific amount of weight that's individualized to each person's strength. And we have them dorsiflex their foot towards their head for a 2-minute exercise period, that's shown here. And then we have them stop exercising. And actually, the thing is fading out, so I'll use my own. So we have them stop exercising, and we look at the recovery kinetics of phosphocreatine. And the recovery depends on ATP generation by oxidative phosphorylation in the mitochondria. And this has been shown by other investigators that it can serve as a marker of mitochondrial function. So going into this technique, we actually had hypothesized that because wild-type p53 promotes mitochondrial function, that individuals with a mutation in p53 may actually have diminished or decreased mitochondrial function. But to our surprise, we found that the individuals who carried the LFS mutations actually had a faster or shorter phosphocreatine recovery time constant compared to carrier individuals, as shown here. And this suggested that they actually had improved or better mitochondrial function compared to, again, the control non-carrier individuals. So we further confirmed this using blood cells as well as muscle myoblasts obtained by needle biopsies. And essentially, we were able to show that there was increased oxygen consumption in the individuals that were carriers compared to the non-carrier individuals, both in the basal and the stimulated state of respiration. This is whole cell mitochondrial function. We're able to also show that the TFAM and the SCO2, the p53 target genes, were also increased in some individuals who carry the specific LFS mutation shown here. This is just protein levels compared to the non-carrier individuals here, shown on the left side, and also another hotspot LFS mutation to 273 histidine mutation. Actually, okay. So we further substantiated this finding by turning to the LFS mouse model that you've already heard about. This is the 172 histidine knock-in mutation. This corresponds to the 175 mutation in humans. And we're able to show in a treadmill exercise test in mice that you can see here a mutant allele dose-dependent increase in treadmill endurance in these mice. I should also mention that these mice, the LFS mice, really are morphologically, developmentally identical to wild type, so it's not like they're bigger or smaller, that kind of thing. But anyway, we're able to show this, again, this mutant allele dose-dependent increase such that the ones that had 2 copies of the LFS mutation actually had almost twice the endurance exercise capacity of the wild-type mice. We prepared mitochondrial and looked at oxygen consumption from skeletal muscle of these mice, and again, we're able to show a mutant allele dose-dependent increase in respiration as shown here. So we think that this increase in mitochondrial function in LFS can be explained by at least 2 different mechanisms. And one is if there's dissociation between the cell cycle and the metabolic activities of p53, and I think this was also nicely demonstrated by Wei Gu in his classic, the mutant that retained antioxidant function. And we can see here that in mouse embryo fibroblasts, both at the RNA and at the protein level, that there was an increase in the 2, the favorite mitochondrial biogenesis genes, SCO2 and TFAM, were increased again in a mutant allele dose-dependent manner, as shown here, while there was, again, complete loss of the P21, the prototypical wild-type P53 target. So again, you see loss of cell cycle control, but there's this increase in mitochondrial function. And this is also reflected at the protein level, as shown here. Another way that we think that this phenomenon could be explained is if p53 has direct effects in the mitochondria. And, and in this regard, we identified a pathway, a specific molecular pathway by which p53 can be pulled into the mitochondria. And this uses this import system called the disulfide relay system. And essentially, it's a relay of electrons from the substrate, which typically is imported through the outer mitochondrial membrane through a complex called TOM. And the typical substrate has multiple cysteines that are capable of forming at least 2 intermolecular disulfide bonds, which p53 also has. Interacts with a carrier protein called CHCD4, first described in yeast system. It's a miR40 protein. And essentially, it's a relay of electrons from the substrate all the way to molecular oxygen that utilizes the respiratory complex for the cytochrome C oxidase. So you can see immediately that this is a bioenergetically driven process where active respiration pulls p53 into the mitochondria. This is just simply a visual representation of this interaction between p53 and the import carrier protein CHCD4, which I should have mentioned that this system is critical for import of a large number of other proteins, not just p53 into mitochondria. You can see that in respiring cells, this is a SCO2 wild-type cells, you can see this nice interaction between p53, which is in red, and CHCD4, which is in green. When you merge these two colors together, you see this kind of merged the yellow signal, which is of nuclear, mitochondrial localization. And the SCO2 knockout cells, which absolutely have lost their respiration, you see this complete segregation of p53, which is mostly nuclear, and CHCD4, which is in the mitochondria. We're actually able to show that this partitioning actually can subcellularly target p53 activity between the mitochondria and the nucleus, and that the mitochondrial fraction appears to can promote mitochondrial DNA repair, which I just want to quickly share with you here using a p53 knockout cell line. If we introduce into a knockout cell line, if we introduce the LFS mutant protein, the 175 again, you can see that if we damage mitochondrial DNA with hydrogen peroxide and then look at the recovery after 6 hours, so 0 hours is 1 hour of damage, that the integrity of the mitochondrial DNA, which is done by a PCR assay, is actually increased with time, and that by introducing CHCD4 exogenously, you can actually accelerate the repair. So by pulling more p53 into mitochondria, it seems to be important. Actually, and even more importantly, we're able to show that in mouse tissues under normal conditions, just normal in vivo basal conditions, that looking at different tissues such as skeletal muscle, liver, and heart, that the integrity of the mitochondrial DNA is maintained in the LFS homozygous knock-in mice, which is in stark contrast to the p53 knockout mice in gray shown here. So you can see that p53 knockout, which don't have p53, actually have less intact mitochondrial DNA, which I think very nicely demonstrates the important distinction between a p53 null state and the p53 mutant state. So we believe that both wild type and LFS mutant proteins that we've studied are important mediators of mitochondrial biogenesis and function both through nuclear target genes as well as its direct activities within the mitochondria. It can regulate multiple processes, including oxidative phosphorylation, that can result in a very dramatic phenotype, at least in the mouse model, such as exercise capacity. But at this point, when we did our studies, the effect on tumorigenesis was less clear, which we have now addressed, or tried to address. So one of the things that we observed in the course of our studies of the LFS mouse, whole mouse metabolism studies, was that the LFS mice appear to oxidize more fatty acids in the mitochondria. And that's reflected by this, what's called this respiratory exchange ratio, the RER ratio. So these are homozygous knock-in mice, and these are wild-type mice. And you can see that the respiratory exchange ratio is lower, and that indicates that the mitochondria is preferring to use fatty acids versus glucose for oxidation. And this was further supported when we did metabolomic studies looking at this acylcarnitine species. These are fatty acids that are imported into mitochondria for oxidation. You can see that the homozygous knock-in mice had higher levels of these substrates, indicating that there was increased activity, compared again to the wild-type animals and the p53 null animals here. And this was interesting to us because there's, again, growing evidence that mitochondrial metabolism, including fatty acid metabolism, may be important contributors to tumorigenesis. And so we wondered whether we could actually alter or try to attenuate this increased mitochondrial function in LFS mice and see if we can affect de novo tumorigenesis. So we turned to two mouse genetic models of mitochondrial dysfunction. And this is the myoglobin deficient or knockout mice and the polymerase gamma mutant mice. We actually became interested in the myoglobin knockout mice because of the potential role of p53 in regulating mitochondria is important for intracellular oxygen species, intracellular oxygen homeostasis as a way of controlling oxidative stress. And in any event, myoglobin knockout mice are essentially phenotypeless. They develop normally, and it's only when you stress these animals to the very maximal exercise capacity that you can see like a 10% difference between knockout and wild-type animals. And as a more direct way of altering mitochondrial function, and we think that might be due to oxygen homeostasis, availability of oxygen for the mitochondria. And then as a more direct way of disrupting mitochondrial function, using this polymerase gamma mutant mice, which are unable to repair damaged mitochondrial DNA. And you can see that the myoglobin knockout mice actually, homozygous knocking mice actually have a higher respiratory exchange ratio, indicating, which is really the opposite of what you saw with the LFS mice, which have a lower respiratory exchange ratio, indicating that these animals are preferring to use glucose versus fatty acids. And similarly here with the polymerase gamma mutant mice, you can also see that they have a higher respiratory exchange ratio, again, indicating generally dysfunction, relatively attenuated mitochondrial function. So we crossed these mice, genetic backgrounds, into the LFS mice. And this slide is just to remind us that we now turn to the thymic tissue because most LFS mice develop thymic lymphomas. And this is just to show you that, again, myoglobin is highly expressed in skeletal muscle where myoglobin is needed for oxygen transport. But it's also expressed in thymic tissue. In fact, it's higher in the LFS homozygous, the homozygous mice compared to wild-type mice as shown here. And we think that, again, that might have to do with increased oxygen utilization in the thymus. Now, by crossing these two different mutant mice into LFS background, we're actually able to show that in the thymic tissue, oxygen consumption could be decreased, normalized almost to wild type. This is 1.5-fold. This is LFS over wild type. We're able to normalize it by introducing the myoglobin knockout on top of the LFS background. And in the polymerase gamma mice, you can see one allele and two allele mutant mice. In a dose-dependent manner, we could actually also decrease oxygen consumption in these mice. And when we measured the plasma lactate, which is a marker of glycolysis, there was actually an increase in lactate levels, plasma lactate levels, again in a dose-dependent manner, at least for the POLG mutant mice. And we think this could be a compensatory increase in glycolysis secondary to a decrease in mitochondrial function. So the important question is, by altering metabolism in this way, can we affect cancer outcome? And the answer is yes. We see that these control mice are actually the LFS homozygous mutant mice. This is looking at cancer-free survival in these mice. And we're able to show that both for the myoglobin knockout as well as the polymerase gamma heterozygous mice, there was a 40% increase in cancer-free survival in these mice. And in the polymerase gamma homozygous knock-in mice, this was about an 80% increase in tumor-free survival in these mice. Now, the polymerase gamma homozygous knock-in mice have a very severe phenotype. It's actually used as a model of premature aging. And we are much more excited about this, the myoglobin knockout and the polymerase het mice, which again have essentially no phenotype and have normal lifespan. And so by just this mild disruption of mitochondrial function, we're able to improve survival in these mice. So that raised the possibility that potentially pharmacologically, that this kind of mild attenuation could be achieved pharmacologically, which brings us to Fazana's metformin study that she very nicely outlined, and so I don't need to really even introduce metformin. And essentially, I should give her a lot of credit for really making this collaboration possible, which I'll actually describe. But we first wanted to test this in the mice. And so we treated LFS mice with metformin. And again, I don't have to say much about this drug, other than that it can inhibit respiration, as Fazana mentioned. And so we treated these LFS mice with different doses of metformin in drinking water. So this would be a survival study long term. And we're using a dose of metformin, 1.2 milligrams per mil, which has been shown by other investigators to result in plasma levels of metformin that is found also in human, at the human doses, such as the 2,000 milligrams that Fazana mentioned. So you can see that in a dose-dependent manner, we can inhibit oxygen consumption in the thymic tissue, again, looking at just oxygen consumption in the thymus in this manner. And importantly, we're able to show that by treating these mice long-term, at the higher, at least at the therapeutic dose of metformin, that we have a modest but significant increase in cancer-free survival. This is about 22% increase in tumor-free survival. And I could wave my hand and say, why is this not as significant as the genetic model? But we also have to remember that these treatments are starting later in life for these mice because during in utero and during the weaning stages, we can't feed these mice with metformin per animal protocol. And so we have also, as I mentioned, collaborated with Fazana in her clinical protocol to obtain samples and follow up some of these patients to look at mitochondrial function. And I just want to very briefly share with you this work. Again, here's the outline of her treatment regimen. So essentially, we're looking at mitochondrial testing pre-treatment at week 8 and 14, which represent the maximal dose that was able to be achieved. And then there's a washout of 20 weeks where the metformin is now taken off. And we're very happy to see that, indeed, if you look at the oxygen consumption of the mononuclear cells isolated from blood from the study participants who are taking metformin, we're able to show this decrease in mitochondrial respiration in cells. We're just measuring in 2 different ways. And during week 8 and week 14 during treatment, and then during the washout phase at 20 weeks, you can see that there was recovery of oxygen consumption with the drug washout. And importantly, we also wanted to, again, look in vivo under normal conditions, because these are all ex vivo experiments where cells are taken out of, purified from the blood. We used, again, the phosphorus 31 MR spectroscopy, looking at in vivo marker, again, of mitochondrial function. And we're able to show that at week 8 and week 14, compared to baseline week 0, that there was a delay in phosphocreatine recovery time constant. And this is for each individual subjects. That was significant, and this became non-significant after the washout period of 20 weeks. And I should mention that this, again, we're looking at imaging of mitochondrial function in skeletal muscle. And this is very encouraging because it's important to know where this drug is having effect. And as it was earlier mentioned by multiple investigators, rhabdomyosarcoma is a common LFS tumor, and this is skeletal muscle mitochondria that we're measuring. So I wanna it's just my summary. So by starting out with our exploratory studies, looking at very basic mechanisms of P53 regulating mitochondria, we've been able to go on to show in our LFS mouse model that by modulating mitochondrial function, that we can indeed affect cancer outcome, and that is also feasible to do this at least by those markers I showed you in patients with LFS, and I hope that can help set sort of the platform or the stage to conduct a larger placebo-controlled study, I think which would be necessary to show this, because these are all, again, model systems that really we have to be very careful interpreting this data. And again, I'd like to thank everyone who really made the study possible, the LFS participants who just incredible amount of effort by the patients to go through this, to help us with these studies. All our collaborators that helped make this possible, and both Fazana Walcott and Louis Strong that really helped early on. Stop. Thank you.